So today is just a demo, like what we are going to do in the next 6 months, or maybe just before the exam, what we are going to do. Just we will discuss the basic things about. So since ASRB, they have given syllabus, right? So I divided the syllabus into different chapters for our easy convenience. We will go chapter wise, and as well as I have also divided into topic wise and as well as concept wise for our easy understanding. So we'll always starts with where this plant pathology started, first one, and later we'll slowly go and get into history. So here I have divided this history into different areas. There is a reason behind this. For example, if you see the previous year question papers, starts with 2006, 7, 9, 14, 1, 14, 2, 18, 1, 18, 2, 19, 21, 23, all the exams solidly we will get at least 8 to 10 questions from the history of plant pathology. They will ask basic questions only. So at the end we do also have this previous year discussions. So I will show you how important this history is, like from mycology, bacteriology, virology. I have divided into different areas. So by reading this history we will get a solid amount of questions, at least 6 to 8. So I have divided into different sections in detail contributions about each and every single scientist we are going to see. So for this contribution also I have pointed out like only the required points and the important points only I have pointed out. And coming to the next one, in general plant pathology, general aspects of plant pathology, what are the different groups of plant pathogens are there, and all those things we are going to see in general plant pathology. In mycology, since it is mycology is a very vast area, so I have sorted out like the only important things what we need to discuss starts with the general characters and the somatic structures, sexual and asexual fruiting bodies, and some couple of important life cycle we will see. The next one is fruiting bodies, taxonomy and classification. In classification, we are going to see in detail about every single class, phylum, order, and up to genus level we will see. And then you can ask me, sir, within the short time, can we cover everything? Of course, we can cover, because I have prepared a notes very crispy way so that it will be very easy to to read and to understand. So all the important phylas like Jagomycota, Ascomycota, the days I have divided. For example, Ascomycota is a little bit bigger, right? So I have divided. I have given into 3 or 4 days. For Basidiomycota anamorphic fungi. Another important thing is at the end of every week there is a weekly test. End of every day daily test will be there. For example, we are finishing general plant pathology today, and the next day, so after class I will send you a question paper. Weekly test will be in maybe Google Sheets, but daily exams will be conducted through by sending question papers, and we also discuss the answers related to the particular topic. And another thing, every month, at the end of every month, monthly test will be there. This is a very important one. This test will be conducted for 2 days. The first day exam and the second day evaluation. This test must be conducted in Google Forms only. You must attend the exam so that we will let you know at what situation you are at. In that way we will also train you, because we never know that at what situation you are at, whether you know basics or whether you are in immediate in intermediate range, or at what situation you are at. We will let you know, then only we can train you in that dimension. So just try to attend the exam. At monthly exam, it's a very important one. And in the bacteriology, different things will be there. This is just what kind of things that we are going to discuss, and all those things I have sorted out, just general introduction only. Okay. And another thing, then you guys will ask me like, sir, whether it covers with the syllabus that was given by ASRB net or SMS STO. Of course, yes. So we will cover all the things. If there is any leftover topics, those who have registered, you can ask me. I'm very happy to share the new topics. If you want, we can also discuss the new topics. And weekly tests, monthly tests, everything at the end of, for example, if you are discussing applied plant pathology, fungicides classification based on their chemical nature, formulations, and all those things will be there, right? So at the end of everything, weekly test will be there, which means, for example, if you are finishing this fungicides and classification, on the next day we do have an exam on this fungicides and classification. On the second day we have a class on chemical nature. At the end of this class, on the whatever we have discussed the before, there is an exam, and again the next class it will continues goes on. At the end of the week, weekly test will be there. That is also very important one. If you attend, then only we will let you know. For example, don't worry, like you might be in a situation like you just started plant pathology, you don't even know, might be the basics and all those things. Don't worry about it. For this 6 months or maybe 1 year, whatever you are registered for, we will train you until the last day. until the last day classes will be taken, until the last day exams will be conducted, until the last day we do evaluation. And one more thing is like after weekly test, yeah, and the most important thing is majority of the people are not that much good at two aspects. One is the laboratory techniques and methodologies especially, right? So we have given very few only, but we have sorted out many other things, but just for our sample understanding I have given this. Starts with the PCR, lamp, ELISA, and so many things are there. So you might have studied the basic things about PCR. So you know PCR, polymerase chain reaction, like maybe Carey Mullis 1983, basic things you know. So how many types of PCR are there? If anyone can tell, how many types of PCR are there? Now total types I'm asking. Can anyone can answer? Yeah, anyway, no problem. Yeah, someone raised your hand. Can you? eh, Yeah, anyway, I'll tell you. There are around 40 types of PCR are there. There is a small story behind this, the discovery of PCR and the different kinds of tag polymerase. Again, this thermos aquaticus, there is lots of stories behind it. Then you can ask me that what we will get if you go through all those things. Remember, we are not only training for net, you are also training to be a good plant pathologist for the future as well. Because, after net, just imagine this net is a preliminary or the basic test for SMS, STO, ARS, everything. So if you know all those things, who knows, ARS will be, like we can consider it as agriculture, UPSC, right? So they will ask in detail different kinds of things they will ask. Who knows, you might have cleared ARS in an entrance exam, they might have asked you different things, right? So one of my friend who attended, a couple of years before, they have showed you a picture. The picture is Vanderplank. He know everything about planter plank, what all the contribution he have done, he know everything. But when he showed the picture of the Vander plank, he couldn't able to identify the picture. That way also they will ask the questions, because they can ask questions from anywhere else. We will prepare from all the directions, so that you can see it is not just about net only. Because who knows, maybe after finishing that, you might be in a assistant professor in somewhere else, at least to tell you or to teach you to your students. So having the overall knowledge is very very important. That we will provide you, that we will train you as a good plant pathologist. The most important thing is physiological and molecular plant pathology. Here I have given a couple of things, and the most important thing we need to take a consideration into the plant defense mechanism, that to host pathogen interaction. How this host pathogen, if you understand this host pathogen interaction, how this host will respond, how this how this pathogen infects, and what are the different kinds of origins can get activated when the plant get infected, like this damp, PTA, ETA, all those things are as a part of host pathogen interaction, right? All those things that we will discuss in detail about. I just want to make sure this molecular plant pathology to be elaborated this time, because last time we have conducted a free training on this for 50 days. In that time we have not explained in detail about this molecular physiological plant pathology. But this time we will go in detail about every single aspect. At the end of every single aspect, we will also discuss what are all the probability questions. If there is any doubts, you can also ask me. We will share the ideas. Yeah, this is about quarantine methods also. There is no separate chapter for quarantine. Generally biosecurity or biosafety, something they will say. So what are all the different kinds of rules and regulations are there? Another important thing is this phenerogamic plant parasites. There are only four are there, like Cuscuta, Loranthus, Warabank, and Striga. Again, if you take a look into the previous year question paper, solidly we will get at least two questions, at least. So if we combine this one question, two question, three questions from the different chapters together, it will come at least 80 to 90 questions. If we are well versed in this, the areas what we are doing, or what areas what we are well concentrating, just imagine. So hope you guys know very well, for general category it is 50%, which means out of 150 questions, if you answer 75 right, that is more than enough. Just imagine. So in that way you can calculate yourself. Since you are writing for the, if you are writing for the first time, don't aim for 150. Okay, just answer 90 to 110, depends on the strength and the difficulty of the questions. Don't go beyond, because we know very well negative marking is there. We also take a look into that. Okay, I wrote net for 5 times, so I have very good experience in this net, and how to write, and how to get out of this net for the first time. So don't worry about it. You might have written maybe one times or two times. It is not it's not about to clear immediately. Sometimes it will take little time also. I know few of my professors, they are writing, they wrote and still writing from past 8 years, still they couldn't able to pass. But that doesn't means they are worthy, they are not worthy. So there are many factors that affects the kind of preparation that you are doing just before the exam, or you may start the preparation right now, or maybe you may leave it in between, or at the end, or just before the exam you might be busy in somewhere else. Various factors will be there. So such kind of people who can't able to spend the time also, we have another idea for last minute preparation. We will train you very intensively for 1 week. You will put everything out and just concentrate on the net. We can assure you there is around 70 to 80% of the chances of clearing net. We already prepared a last minute preparation, so that will be greatly helpful for you. And another thing is like discussions. So once we finish this all these things, so we have discussions on the previous year net question papers. We have around 10 to 11 previous year question papers: 2006, 7, 9, 14 1, 14 2, 14 18 1, 18 2, 19, around 21, 23. So not just discussions, we will we will wind up all the topics and whatever we have discussed previously, we will merge together. Like for example, if you are studying something, bodo mixture was discovered by Miladet. Yes, this everyone knows, even a UG student will tell, even my nephew will tell this. But the thing is, since we are preparing for some kind of exam, you need to recall every single point that is associated with bodo mixture. If I tell you bodo mixture, you will start with okay, Miladet. So this bodo mixture was developed by Miladet in 1882 to 1885. Actually that was finished at 1885. Okay, why this bodo mixture was developed Miladet? This is especially meant for grapes downy mildew. What is this grapes downy mildew? Okay, it is one of the epidemic in 1878 in France. Okay, 1878 year is also important. Okay, in 1878 it is an epidemic. So what is the pathogen? Because the pathology is all about studying of pathogens, we must recall the pathogens, right? Okay, grapes downy mildew, Plasmopara viticola. Okay, what Miladet had done? So he discovered a specific fungicide called Bodo mixture. Why he named Bodo? So Bodo is the university name where he worked. Another important point is Bodo is also the a specific area name where he discovered this mixture. That's why they named it as Bodo mixture. Okay. So what is the composition? Maybe 1 is to 1 is to 100. What is 1 is to 1 is to 100? 1 kilogram of copper sulfate, 1 kilogram of lime, 100 liters of water. This UG student will know. Let's dwell in deep into it. The original composition that was developed by Bodo mixture, the Miladet, is 5 is to 5 is to 50. 5 lb of copper sulfate, 5 lb of lime, 50 gallons of water. What is 1 lb? It is around 0.4 kilos. Around 5 lb means it will come 2.26 kilos. So what is 50 gallons? 1 gallon is equal to around 3.6 to 3.8 liter. If you combined in that way also, you will get 1 is to 1 is to 100. So I'm generally telling this is the way how we need to prepare. In this direction we will train you very well. So other leftover topic, if there is anything else also, we will discuss. And daily exams, this is a very very important one. However you are writing the net exam, for example 150 minutes, 2 hours, just before 1 month, every single day, not one exam, we will conduct 2 exams, 2 sessions, we will conduct the exam, we will discuss the things. If there is any doubts, you can ask me. However you are preparing, however the Usain Bolt will prepare just before the sprinting, we will train you in the same way, exactly to the same fashion net exam. Some people mailed me regarding, like for example, if you are taking 6-month membership, this will expire just before the ARS exam, can you train for ARS also? Of course I will train you. See, 15 days, 20 days, it is not a big problem. Even though you are registering for 6 months, we will train you 7 or 8 months, no problem at all. So based on the requirement, if the ARS exam is at the December, your membership might expire at November, but no problem, we will train you free for 1 month, no problem at all. And the last 1 week we will give you the basic understanding and training about the ARS. Whoever are preparing for the ARS, we will give you basic training. There is nothing to worry about it. And there is slight changes in the in the schedule that I have given. According to the time, we will slightly changes, not too much deviation, slightly changes. And couple of people asked that, I do understand that maybe this is the time to submit many of your thesis, maybe masters or maybe PhDs. I do understand your time concern. Many people are asking like, sir, can you provide the recorded lectures? Of course we do, every single day, but not we do upload to the YouTube in a private mode, because not everyone can watch our videos hereafter, because you are paying so that you only can watch. For example, you are registering, make sure that your email ID is right, because we will send you the link to your email ID. Whenever you are free, open the link, you can watch the video. And the thing is, we are providing the recorded lectures, there is nothing to worry about it. Yeah, for your understanding, this is demo one, right? A sample discussions how this training is going to be. I randomly selected. Okay, generally Pythium and Phytophthora I randomly selected. We will discuss something about this. Okay, we'll start with this Pythium. Generally, the meaning of Pythium is decay, which means Pythium means decay. Generally, this Pythium causes diseases at mainly at nursery stages, especially this damping of disease. Especially we can mainly observe on nurseries, right? Pythium damping off or damping off is mainly caused by Pythium aphanidermata. Majority of the damping off, maybe one or two damping off, it is also associated with Phytophthora. See here the Pythium produces sporangia because this Pythium belonging to Wumicota group of fungi, right? See here I'm generally telling you how this training is going to be, because if we are discussing in a topic wise, we will go in detail about by discussing the taxonomic rules and regulations along with this general characters and all those things. Generally I'm telling. So then you can ask me, sir, these points are enough for exams? You will come to know at the end of this PowerPoint. Anyway, generally causes damping of diseases, especially nurseries. Generally Pythium is decay. So Pythium is generally a Wumicota group of fungi. Okay, so generally this Pythium produces sporangia because Wumicota group of fungi produces sporangia, right? The sporangia is generally globose structure. This is simply like a round or globose structure, which is a non-papillate. Another, you have a doubt, what is this papillation? Papillate, non-papillate, semi-papillate, three things will be, I will show you. Yeah, this is small outgrowth or some kind of constructions will be there at the end of sporangia. This is sporangia. Inside sporangia, what will be there? Sporangia will be there, right? So there is small kind of construction. This is called papillate. If it is raised a little bit more, it is called papillate. If it is half raised, it is called semi-papillate. Okay. If it is, there is no papillar, there is no outgrowth, it is called as a non-papillate. When it comes to Pythium, it is a non-papillate, which means there is no outgrowth above the sporangia. Okay. And the sexual spore, all Wumicota group of fungi produces sexual spore as oospore, asexual spore as zoospore. Male reproductive organ is called antheridium in Wumicota, female is called oogonium. In the meantime, Pythium also produces male and female reproductive organ. Male is called antheridium, female is called oogonium. Sexual reproduction is gametangial contact type in Pythium. For example, if you see the Pythium life cycle, the sexual reproduction is gametangial contact type. In Wumicota, generally the sexual reproduction is gametangial contact type only. So you just listen this. At the end, I will tell you how we need to divert into exam orientation. Okay. Paragynous type of antheridium. As I told you, what is antheridium? Antheridium is a male reproductive organ, is called antheridium. So this antheridium is of paragynous type. Again, new terminology. What is this paragynous? Very simple. Male is called what? Antheridium. Female is called oogonium. Generally, male is smaller compared to female. Okay. This male and female which lies side by side, if you see here, this is paragynous, this is antheridium male, this is oogonium female. So if the male and female which lies side by side, it is called as paragynous type of antheridium. Okay, it forms a vesicle above the sporangia in the life cycle. This is a special feature, very important. If you see in any life cycle, there is no vesicle formation takes place. For example, if you see, this is sporangia, right? Above the sporangia, there is another structure will be formed, especially in Pythium only. This is called vesicle, where zoospore formation takes place in vesicle, not in the sporangia. Okay, so in exams they will ask formation of vesicle can be observed in the life cycle of which of the following pathogen? A, Pythium, Phytophthora, Fusarium, something like that. There is no other life cycle where the vesicle is produced above the sporangium except this Pythium. This Pythium produces a vesicle above the sporangium. This is sporangium. Above the sporangium, small another structure will be, that is called vesicle. Inside the vesicle only, this compartmentization or zoospore formation takes place. That is called vesicle. Okay, most of the Pythium species are homothallic. Homo means both male and female organs are present, right? Hetero means both male and female organs are present on different thalli. Homo means same thal, like that. Okay, so here for the management of Pythium diseases, what we do recommend is a metaloxyl. Generally, it is a specific fungicide for Pythium diseases. So the trade name of metaloxyl is Redomil. So UG students generally studies the metaloxyl, but since we are in PG and PhD, we must study this trade names also. The specific fungicide for Pythium diseases or Wumicota diseases is Redomil. So if the metaloxyl, sometimes metaloxyl are available in combinations, metaloxyl plus mancozeb. So if this two combinations are there, the trade name of this metaloxyl or mancozeb combination is Redomil Gold. So phosphatyl aluminum is also, sorry, phosphatyl aluminum is, yeah, just a minute. Yeah, phosphatyl aluminum is also recommended for Pythium disease. And another important point is we do also recommend Bodo mixture. Again, there is a new terminology. What is this Bodo mixture? Yeah, I'll show you. Yeah, This Bodo mixture is otherwise called as generally holy water. This was developed by Millardet in 1882 to 1885. Again, why this Bodo mixture was developed by Millardet? Especially, it is recommended for a disease called grapes downy mildew. Grapes downy mildew is caused by Plasma parviticola. It is an epidemic in France in 1878. What is the exact composition of this Bodo mixture? 1:1:100. But original composition that was given by Millardet is 5:5:50. What is this 1:1:100? 1 kilogram of copper sulfate, 1 kilogram of lime, 100 liters of water. This is generally 1:1:100. But the original composition that was given by Millardet is 5 lb of copper sulfate, which means 2.26 kilos. 1 lb equal to around 0.4 kilo. It will come close to, I think so. So 5 lb means it will come close to 2.26 kilos. Hydrated lime, again 5 lb, which means 2.26 kilos, and 50 means 50 gallons of water, not 50 liters of water, it is 50 gallons. 1 gallon equal to 3.8 liters. Around it comes around 160 170 liters, it will come close to, right? If you divert or convert this formula, again it will come close to 1:1:100 only. Why confusion? That's why the scientists later changed it to 1:1:100. Now let's discuss here what are all the important points. Every single point here mentioned is an important one. I will tell you how. In exams they will ask damping of generally affects, or damping of disease is generally caused by which of the following pathogen. So damping of generally caused by Pythium only. Damping of disease of, or Pythium damping of generally majorly infects which of the following crops? A, vegetables, cereals, maybe cash crops, some other. Generally this Pythium or damping of disease is very severe at vegetables only. Again they will ask the shape of sporangia that is produced by Pythium. Pythium produces shape globospora, right? So male reproductive organ of oomycota or Pythium is called antheridium, female is called oogonium. Or sometimes they will ask what is the kind of sexual reproduction that is observed in Pythium. So all oomycota group of fungi, this Pythium also gametangial contact. Again another question, what is this gametangial contact? So male, this is male and female, they come in contact with each other and there is a small fertilization tube is developed. So this in detail about this we will discuss in sexual reproduction. This fertilization tube generally helpful to transfer this nuclein from male to female. Their reproduction takes place and the oogonium forms, right? So here, sorry, the zygote forms. So here the gametangial contact type is the specific kind of reproduction or sexual reproduction that we observed in Pythium. So another paragynous type of antherium. This is also important point. What is the type of antherium that is produced by Pythium? Or paragynous type of antherium is produced by which of the following fungi? Pythium, Phytophthora, Albugo, or some other thing. Generally, paragynous type of antherium is produced by Pythium. Okay. And another important, very very important point. This question was asked very long back, not only in NET but also various other kinds of exams. So formation of vesicle above the sporangium can be observed in the which of the following life cycle? Pythium, Phytophthora, they may also ask other things. Okay, around what is the specific fungicide recommended for Pythium? Metaloxyl. The trade name of metaloxyl is Ridomil. It is very important. The trade names is also very important. They were asking. Yeah, this is about something about Pythium and couple of important diseases. Maybe sometimes they also ask like pathogens also, right? So since we are discussing, let's discuss every single probability topic or probable point related to this Pythium. So damping of a vegetable generally caused by Pythium aphanidermata. Sometimes Pythium divarianum is also, but for UG level and PG level, we do say like generally damping of is caused by Pythium aphanidermata. So when it comes to PhD, everything we do see in a research orientation, that might be different, but generally damping of vegetable is caused by Pythium aphanidermata. So for example, if you see foot rot of papaya, it is also Pythium aphanidermata. This question was asked. I will show you. Rhizome rot of ginger, very very important one. Rhizome rot of ginger is caused by Pythium aphanidermatum, and there is another species called Pythium myriotalium. And fruit rot of cucurbits, Pythium aphanidermatum. Damping of mustard and beet, Pythium mammillatum. And damping of general angiosperms is caused by aphanidermatum, dibarianum, ultimum, irregularly. And sometimes they may also ask for pathogens, right? That's why I have given this. And we have discussed about Pythium. Let's see some important points related to Phytophthora also. See, this is just a demo only. Here I might have represented maybe 2 to 3 slides, but when it comes to daily presentation or daily lectures, that might be different. Every single slide I am just trying to put couple of probability questions and previous year combinations and all those things. That will be greatly helpful, right? Yeah, coming to this point, Phytophthora. Generally this Phytophthora, which means Phyto means plant, Thora means destroyer. Okay, Who called this Phytophthora as plant destroyer? Anton de Berry. Who is this Anton de Berry? Maybe father of plant pathology. Not maybe, he is father of plant pathology. So in that way, we'll see. The first one is generally, this is the pathogen which is responsible for Irish famine. Why it is called Irish famine? Because the disease first happens in Ireland. See, what we know is the disease first happened in Ireland. That was not true. The disease first happened in USA, Mexico, various countries were there. Actually, the pathogen was first reported from England and this Wales is around 1842, but the severe epidemics happened in 1845 only. This is the Anton de Berry was the not first person who studied this. There are plenty of person. In 1842, there is a scientist called Van Moren. He is the first person who described and studied this pathogen, but nobody found the exact reason behind it. Many scientists were, they were actually, fought for this, but no one completely identified what exactly is. Finally Anton de Berry came and he have done some contribution that is different anyway. So this Phytophthora associated with Irish famine, we can also call as late blight of potato. What is this late blight of potato? Is caused by Phytophthora infestans. What is this phyto means? Phyto means plant. Generally plant pathology we can also call as phytopathology, right? Okay, phyto means plant, thora means destroyer, infestans means it causes a devastating infection. That's why they named it as Phytophthora infestans. So the name Phytophthora means, just now I told you, plant destroyer. This was one of the question. The name Phytophthora in Greek language, generally this Phytophthora in Greek language, in Greek terminology, it means plant destroyer. In one of the exam, not in net, maybe in entrance exam, I think, when I was writing 2016 Jane KVV, I think so. So in that year they have asked this question, like Phytophthora generally means plant destroyer, plant killer, plant, some kind of things they have asked, but still I remembered this question. So phyto means plant, thora means destroyer, which means it is destroying the entire plant. That's why Anton de Berry only named this Phytophthora as plant destroyer. Okay, it is a well studied, well suited name because it is destroying and killing the entire plant. Okay, so generally the name Phytophthora infestans was given by Anton de Berry. Why I have given 2 years here? In 1861, he gave the first proof this Phytophthora infestans is the reason behind this lead blight of potato. Before that, there is very big history and mystery is there. I can't able to tell you right now because again it will go very lengthy one. So anyway, Phytophthora infestans, the name was given by Anton de Berry. In 1861, he discovered the life cycle, but he only gave the term Phytophthora and named the pathogen as Phytophthora in 1876, which means two things will be there. In 1861, he discovered the life cycle and he gave the proof that lead blight of potato is caused by fungi, but he never named the pathogen. And 1876 only he named the pathogen as a Phytophthora infestans. Then you can ask me another question. So he discovered the life cycle in 1861. Then before that, what is the name of this lead blight of potato? Before it is called as Botrytis infestans. Okay, the name Botrytis infestans was given by Montag. Who is this Montag? He's one of the French retired army person. He is not a mycologist. He is an army surgeon. At that time there is no special branch of phytopathology or botany and all those things. Everyone will study about everything. So generally this Montag is a surgeon. I would say generally he is one of the high professional army person in the Napoleon army. Sorry, not Napoleon army, that was later continued by some other kings later. So he is one of the army general, generally a doctor, I would say. He named the pathogen is a Botrytis infestans. Later the same name was also given by Berkeley. Who is again this Berkeley? He is one of the very famous English plant pathologist. The term mycology, what we are studying, what is mycology? Study of fungi. So the term mycology was given by Berkeley only. Again in detail about this person contribution that will discuss it in the history. Okay, 1845 they named lead blight of potato is caused by Botrytis infestans. At the time nobody believed that a fungi can cause a devastating infection or devastating infestation like this. So people didn't believe that. Ultimately, the Anton de Berry proved this Phytophthora infestans is the reason in 1861, and in 1876 he named the pathogen as a Phytophthora because Phyto means plant, Thora means destroyer, because it is causing a devastating infection. It produces amphigynous type of Anthridium. So again, all the Oomycota produces male is called Anthridium, female is called Oogonium. Again, this Anthridium in previous I will show you. Take a look at this. In Pythium, we have discussed paragynous type of Anthridium was there in Pythium. So when it comes to Phytophthora, amphigynous type. What is this amphigynous? So paragynous means a male and female which lies side by side. It is called paragynous. Para means side by side. What about this amphigynous? See, take a look at this amphigynous. This is antheridium. Above the antheridium, oogonium lies. So the antheridium actually passes, this oogonium actually passes through the antheridium, and they are sitting on above the oogonium. Sorry, they are sitting above the antheridium, that is called amphigenous. Anyway, without confusion, I will tell you this is antheridium. So above the antheridium, the oogonium will sit. Actually, generally it passes through the antheridium and form a globose structure above the antheridium. So if it is sitting above the antheridium or male reproductive organ, that is called amphigenous type of antheridium. This is a very, very, very important point. Hundreds of times this question was asked in various entrance exam. You can take JRF, SRF, various kinds of exams. This question was repeatedly asked. Amphigenous type of antheridium is produced by Phytophthora. Or Phytophthora produces what kind of antheridium? Amphigenous. When it comes to Pythium, paragenous. Sporangia. Here the Phytophthora produces a lemon-shaped sporangia. Exactly, yeah, the sporangia exactly look like this. This is the exact representation of a lemon-shaped sporangia. But when it comes to Pythium produces what kind of sporangia? Globosporangia. But when it comes to Phytophthora is producing lemon-shaped sporangia. So Phytophthora infestation generally for sexual spore is oospore, asexual spore is zoospore. We know very well, right? What is zoospore? Flagellated spore is known as zoospore. Generally, this is how the flagella will look like, right? So again, flagellation that we will discuss in detail later. So generally for the development of zoospore, the temperature requirement is generally 12 to 15, minimum 12. It can also go in between 12 to 15. And when it comes to germ tube formation, it is, or sporangia formation also, it will require around 18 to 20. Okay, Anton DeBerry was the person everything behind this Phytophthora. If you call Phytophthora, we cannot get out without this Anton DeBerry, because this person is everything about Phytophthora. So this is the pioneer person who actually did lots of contribution to this Phytophthora. Let's go through who is this Anton DeBerry, what kind of contribution this Anton DeBerry did. Okay, yeah, who is this Anton DeBerry? We know very well. He is considered as father of plant pathology, also considered as father of modern plant pathology. He is also considered the father and founder of modern experimental plant pathology. So father of plant pathology, because he have done lots of contributions. So why he has respected with the honor of father? See, in couple of books it is also mentioned that J.G. Kuhn is considered father of plant pathology. No, it is not, because he have done, of course, he have done lots of very good contributions, but by concerning the various aspects of plant pathology, we cannot deny the fact of Anton DeBerry that he have done lots of contributions compared to J.G. Kuhn. I will tell you what are all the things he have done, why we need to consider him as a father of plant pathology. So he is also considered a father of modern plant pathology also. He is also considered a founder of modern experimental plant pathology. Why this? For example, if you see, uh, before initiation of germ theory, if you recall the history, right now we are just recalling all the history, what exactly happened in the timeline. What I observed is actually the germ theory of diseases that was first whistleblowed by many scientists, but it was first taken into consideration by Louis Pasteur in 1862. The Louis Pasteur got his idea from Anton DeBerry when he discovered the life cycle of lead blight of potato. He got an idea, which means a fungi can cause the disease, which means there might be a pathogen which is associated just behind any disease. That is the exact reason initiation of this germ theory of disease by Louis Pasteur. So, which means the germ theory of disease was first evoked by Anton DeBerry only. I can also prove you how he have evoked. So I will tell you. So he have conducted an experiment, a simple pot culture experiment, to prove that before that there is no experiments. They will simply, there are lots of group of people who believes in one theory, that is spontaneous. There is also opposite person who don't believe, or still they argue the spontaneous generation theory. But at the time there is no exact experiment. People have no idea about the conducting experiments. So small things might be there, but the true experiments were started by Anton DeBerry by conducting a small pot culture experiment, and proved everyone with conviction that this late blight of potato is caused by fungi by inoculation. So in that time itself we have a very advanced knowledge of the vast area, and the introducing the new terms. So he was the first one who introduced the conducting of experiments. That's why he is called as father and founder of modern experimental plant pathology. And he wrote a book. This is a very important book, comparative. Actually he have wrote many books and lectures. Comparative morphology and biology of the fungi, mycetiosa and bacteria. These are all this is the one of the important book that was written by him. And he coined many terms. Haustoria, what is haustoria? We know very well. nutrient absorbing organ or energy absorbing organ of any fungi. You can take what is the Astoria? Nutrient absorb. What is the other function of Astoria? There is no other function, only nutrient absorption. Why fungi require nutrients? You guys know very well. However, we need food for survival. The fungi also require nutrients. So to get the nutrients, to suck the nutrients, it requires a specialized organ called Astoria. Every fungi produces different kinds of Astoria. Finger like Astoria is produced by Phytophthora. Wedge shaped Astoria is produced by Arctobis. Some other kinds like shape of Astoria also will be there. In that direction also will train you. So generally this Astoria we can also call as sinkers. Symbiosis. The term symbiosis was also coined by Anton DeBerry. What is symbiosis? We know very well. Mutual relationship between two organisms, that may be plant or fungi or whatever it is. So generally mutual relationship between two organisms called symbiosis. The term symbiosis is also coined by Anton DeBerry only. Teletospores, we can also call it teleospores. Mycetiosa, generally this mycetiosa is the term referred to protozoa. At the time there is no word protozoa. Instead of representing protozoa, he initiated another new term called mycetiosa. The first person who studied about sex hormones and also worked on siren and anthraciol and autoecious and heteroecious nature of rust. In 1850s and 60s itself, he was very well versed about this wheat stem rust, and he was the first person who studied about autoecious and heteroecious nature of rust. And chlamydospores, this resting spores is known as chlamydospore. The term chlamydospores is also coined by Anton DeBerry. Just imagine how many new terminologies he have introduced. And his students like MS Voronin, Oscar Brefield, Millardet, HM Watt, these all are the students, and couple of them are did their PhDs and various other kinds of work. In fact, Robert Koch is also trained under Anton DeBerry for couple of days, couple of months, I think so. Just before his death, he's also trained Robert Koch. And one more person, Vinogradsky, we generally consider as father of antibiotics in Russia. But in general, we do consider father of antibiotics is Waxman. But general father of antibiotics is Sergei Vinogradsky is actually from Russia. In fact, this Waxman is also from Russia. Waxman is a student of Vinogradsky. Later, this Waxman, he went to America and there he have done some contributions. Later, become the father of antibiotics by discovering this streptomycin. Okay, right? So, his students he have trained. Here, I have given only 10 here, but just 1 minute. Go down. 5 minutes. Go. Yeah, I'm sorry for the disturbance. Anyway, yeah, students, here I have given only couple. Generally, Anton DeBerry trained over 220 students in his lifetime. The majority of the members become very great contributors to the plant pathology. Here I have given couple of things, couple of scientists, right? Let's study their contributions also. Who is MS Voronin? He discovered the club root of cabbage life cycle. What is this club root of cabbage is caused by Plasmodium brassicae. It is not a fungi, it is a protozoa. It is a protozoal disease, right? So club root of cabbage, in protozoa we do study only one important disease, that is club root of cabbage, which is caused by Plasmodium brassicae. In detail about the life cycle, hypertrophy, hyperplasia, and this life cycle we will discuss in later class, right? Oscar Brefield, he is the first person, he is also a student of Anton DeBerry. He is the first person who developed pure culture technique in 1976 and 77 time, I think 72, 73 time. In the exact meantime, Robert Koch also working on this pure culture technique, but the credit first goes to Oscar Brefield only because he is the first person who introduced this. And single spore isolation is also given by Oscar Brefield only. And now we are using agar as a solidifying agent, right? Before agar, Oscar Brefield is the first person who introduced gelatin as a solidifying agent. This is also one of the important question, right? Before introduction of agar, what is the solidifying agent? Gelatin is the solidifying agent before introduction. The gelatin as a solidifying agent was first introduced by Oscar Brefield only. He is also student of Anton DeBerry. Next one, Millardet. Just now we have discussed, we developed Bodomichere composition is 1:1:100. Grapes downy mildew, right? H.M. Ward, father of tropical plant pathology, is generally considered as, and he have done very good contributions just related to this coffee rust pathogen. We know very well coffee rust is caused by Hemilia gastatrix, and there is another small history. Actually, Voronin is from Russia. It was actually the Anton DeBerry's suggestion who suggested Voronin to go and study this club root of cabbage life cycle, because this club root was very severe in 1870s in a Russia time. So Russian government, they openly offered something like a good price. Whoever will discover and sort it out, we will give a good price. So Anton de may went and discovered it, but it was the original suggestion of Anton de to Voronin, because Voronin is from Russia, so Voronin know very well about Russia. So Anton de told to this Voronin, go and find what exactly it is. It is also another HM ward who actually initiated by who actually sent to Sri Lanka in 1860s, 1870s by Anton de only. At the time, Anton de health was not good. This colonial Britishers, they asked Anton de to find out what exactly the problem with coffee, which is infected with rust, because Anton de cannot able to go because his health was not good. Then he sent to HM ward. There he went and he worked on the aspect of this coffee rust, and he identified very major important things related to this coffee rust, and identified that monoculturing or monocropping is the main reason, something like they have identified. And even you say double G Farlow. So after all contributions, nobody have thought of initiation or introduction of a new course or development of a new branch of science, that is plant pathology, but it was originally G Farlow who started an independent course of plant pathology, that is the foundation stone for plant pathology. So he is also Anton de students. Just imagine how many students he have trained, how many become the greatest contributors. And another thing is, he's Anton de we know very well. You might have saw everywhere, in every single lab. Without his photo there will be no lab. In fact, we are having 5 different photos of Anton de Every single lab is having Anton de right? Astoria, Teltospore, heterosis, Resimba, they're all the terms, right? Another contributions, just now we have discussed, life cycle of Phytophthora infestans, he only discovered, demonstrated the heterosis nature of rust in 1865, which is a very, very important one. Heterosis means, hetero means different. So before, people thought that they have a different kind of theories on this wheat stem rust, because it is the Anton de who blowed the whistle for the first time. This pathogen required 2 different hosts, which is biologically different, not same families, different families. It was Anton de So first physiological work is also initiated by Planton de only in the fungi called Botrytis cinerea, and is also reported the role of enzyme in 1880s itself, just before the 2 years to his death. He also started working on role of enzymes and toxins through direct penetration or tissue disintegration, especially on a fungi called Sclerotinia sclerotiora. Phytophthora means plant destroyer, right? We have discussed. Now we will discuss back. The another important point is, he have done lots of contributions, right? He is not a mycologist. He is originally a human doctor. He submitted his thesis in botany. So actually what happened at the age of 21 or 22, after graduating as a MBBS student or a physician, he practiced MBBS or that medicine for just 6 months. He was very enthusiastic and interested in botany, so that he left everything behind and started working as a mycologist. Later, the rest is history. You guys know everything about it, right? And one another important point is, at the age of 21, when he was submitting his doctoral thesis, he submitted on a specific topic that is actually related to botany. So there is interest started in all those things. Just imagine, a doctor have done lots of contributions in mycology. So anyway, so here in exams, what kind of questions we expect? See, what is the book written by Anton de Comparative Morphology and the Biology of the Fungi, Mosquitoes and Bacteria? This question was asked in 2021 and 2023. I think 23, I think, not 21. Anyway, we have previous year question papers we will go through, and sometimes they may ask by giving one of the term, like which of the following term was associated with Anton de or chlamydospores was the term was coined by, or maybe a heterosis and autosis nature of rust was discovered by. Some kind of questions we do expect. Another thing is, who discovered the life cycle of lead blighted potato? What is the meaning of Phytophthora? Phyto means plant, thora means destroyer. Phytophthora means plant destroyer. Okay, in sometimes we expect like physiological plant pathology work was initiated by Anton de in which fungi? Botrytis cinerea. So now you might have remembered every single line or slide. Just started when we started classes. It's hundreds and hundreds pages, hundreds of information that will come close together. So we need to sort it out in an easy way. So that's what I am telling here. Okay, anyway, generally we have discussed about Pythium and Phytophthora means. So this Pythium and Phytophthora both comes under Oomycota group of fungi. Sometimes in one of the exam, I think 2018, they have asked this simple question. So this Pythium and Phytophthora generally classified under which group? chromista, or we can also call as Wumicota. Wumicota actually classified under chromista group only. Okay, this Wumicota group of cell wall is made up of. This is a very very important point. In the last 5 years, this question was asked 3 times. I will show you previous year question papers. So Wumicota group of cell wall is made up of cellulose plus hydroxyproline. This is a very very important one. In 2020, they have given only one option, hydroxyproline. In 2023, they have given only one option, that is only hydroxyproline, which means Wumicota group of cell wall is made up of cellulose plus hydroxyproline. Remember both. So generally for Wumicota group of fungi, what is the specific fungicide? We have already discussed metalaxyl, right? So Wumicota group of fungi cell wall is made up of cellulose and hydroxyproline, but fungal cell wall chitin we know very well. Yeah, these are all the list of important disease that is caused by this Phytophthora, rubber abnormal leaf fall, Phytophthora palmivora, red gram stem blade, Phytophthora drusellare, this castor seedling blade, very very important one. In history, there is a scientist called J.F. Daster. So he is the first person who is associated with the discovery of Phytophthora genera in India. So in India, Phytophthora genera was first discovered by this J.F. Daster only. That disease is seedling blade of castor, which is caused by Phytophthora parasitica. In exams, they can ask you anyway. I will tell you how. The same question we can expect in 4 different ways. I will tell you one first thing. So J.F. Daster associated with of the following genera Phytophthora. J.F. Daster discovered a disease in the castor. What is the disease name? Seedling blade of castor. So sometimes they will ask J.F. Daster is associated with the discovery of which of the following disease. They will give pathogen name, Phytophthora parasitica, Phytophthora palmivora, Phytophthora aerica, like that they will give maybe disease name. Okay, so the crop is castor. Sometimes they may also ask crop also, right? In that we must train in different directions so that if you have overall knowledge, you can easily answer the question. Tobacco black shank, it is a very very important one. Phytophthora parasitica, Nicotiana, coconut bud rot, citrus gummosis, sapile. Like this, I have sorted out some of the important disease that is caused by this Phytophthora. So sometimes they also ask, like questions from the pathogens or some kind of things. They may also ask. So it is very important to study these things also. Yeah, the difference between Pythium and Phytophthora we have already discussed. I am giving you very few points. What is Pythium means decay, we already discussed. Phytophthora means plant destroyer, right? Phyto means plant, Phthora means destroyer. Who gave this Phytophthora as a plant destroyer? Just now we discussed N20 berry, right? So Pythium comes under Pythiaceae, Phytophthora comes under Peronosporiaceae. Order Pythiales, order here Peronosporiales. In that recent classification, this Pythium is also classified under this Peronosporiales only. Okay. Yeah, another important point, in Pythium both Hastoria and Approsoria are absent. Majority of the Pythium species produces no Hastoria, no Approsoria. Okay, but Phytophthora produces this Hastoria. Okay, and Sporangiospores we have already discussed. It is generally oval shaped or spherical shaped or ovoid shaped, but when it comes to Phytophthora, it is a very peculiar. It is a lemon shaped sporangia. Very very important one. Lemon shaped sporangia is produced by Phytophthora. Okay. Vesicle. So formation of vesicle is a specific feature of this Pythium, because no other fungi can produce vesicle. This is a sporangia, right? Above the sporangia there is another structure formed. Inside this vesicle only juice pore formation takes place in Pythium. Especially Pythium, the juice pore production takes place inside this vesicle only. This is called vesicle. It happens only in Pythium. This is a very very important point. So this vesicle formation takes place only in Pythium. Okay, and another thing is like, yeah, type of antheridia, paragynous type of antheridia. What is paragynous? We have discussed male and female which lies side by side. It is called paragynous. If a female is lies above the antheridium, it is called amphigynous. It is a very important one. Paragynous type of antheridium is produced by Pythium. Amphigynous type of antheridium is produced by Phytophthora. So this is a couple of points. Yeah, this is a previous years. At the end of every single topic, we will try to put some previous year question papers and probability topics also. Okay. So in the 2023, this question was asked: cashew nut damping of pathogen. So cashew nut damping of is general associated with Phytophthora. Okay. Buckeye spot of tomato is caused by Phytophthora. In the Phytophthora, previously I have given buckeye spot of tomato, and it is also associated with some other factors also. Okay. So whom is it is a specific function. See, answer according to the options given. Sometimes the answer might be different what you know, but we must answer according to the options given. You cannot say this is not the right option, I will go with some other option. That is not possible, right? So make sure that our answer must be according to the options. Sometimes So the answer what we know might not be there in the options at that time. Whatever you know, in that way your answer must be close to the options given in the exam. In that way we need to answer. Okay, majority of the time they will ask right exact right questions with the right direct answers only. Maybe one or two occasions with one or two questions might be such way it happens. Anyway, Womycetes specific fungicide. So we have discussed, right, Womycota specific fungicide, metalaxyl. Another point, what are all the different fungi are there in Womycota? Many are there. Two important one, Pythium and Phytophthora. You can also take albugo, downy mildews, all are comes under this Womycota only. So again, in classification we will discuss in detail. This is, since it is just a demo, I'm telling you the basic things. Womycetes specific fungicide, metalaxyl. I think this question is also asked in 2023 only. So another example of amphimobile fungicide, metalaxyl. So metalaxyl having a properties of both acropetal and basipetal, which means upside action, downside action, both actions. So this question was asked in 2021. Okay, there is another fungicide which is also having amphimobile action, that is phosphatyl aluminum. In the option phosphatyl aluminum was not there, then we can go with this metalaxyl. So if the phosphatyl aluminum was there, our first preference goes to phosphatyl aluminum. See, nothing to worry about it. If you keep on attending the classes, you will come to know all those things. First thing. Second thing, we do conduct exam every single day. There you can slowly learn, and our questions are exactly reflecting based on the exam representation only. Not only for net, this training will be helpful for whatever the exam related to plant pathology. Maybe JRF, SRF, whatever, there are hundreds of exams are there in India related to plant pathology. You can use every single point from from the training of us. Another thing, so original bodo mixture composition, we have already discussed, right? The original composition of bodo mixture is given by Melody, is 5:5:50, 5 lb of copper sulfate, 5 lb of lime, 50 gallons of water. That was given in 2019. Phytophthora ramorum causes which disease? Sudden oak death. Yeah, take a look at this. Yeah, we are Phytophthora ramorum, sudden oak death. What is the crop? I might have given here, sudden oak death is caused by, yeah, somewhere it will be there. Anyway, sudden oak death which is caused by Phytophthora ramorum. Anyway, next one, favorable temperature required for zoospore and sporangia formation, 12 and 24. I will show you. Here in Pythium we have discussed this. Yeah, in Pythium, not in Pythium or Phytophthora what? Yeah, sorry, in Phytophthora we have discussed. So generally zoospore formation 12 to 15, gems for the gem tube formation as well as sporangia development in between 18 to 24. In the options they have given one thing, that is 12 to 24. That is exactly reflects, as I told you, sometimes answer might be more or less like that. So we must go with that answer. Cenocytic mycelium, which means no septations are found in the hyphae. Cenocytic mycelium is produced by Phytophthora. Yeah, this is another important point, actually forgot to mention. So this Womycota group of fungi where this mycelium is cenocytic or non-septate or aseptate, which means there is no septa are present inside this hyphae. Okay, what is hyphae, what will be there inside the hyphae, and all those things that will be started from the beginning classes. This is about a demo 1. Demo 2 is much more interesting. I took special reference to rust. There I will portray you what are all the important points. For example, if we are talking about a particular point, we will put you everything about that particular point along with the probability topics also. Everything we will try to portray here along with the same way we will discuss related to exam orientation also. This is a demo number 1. Demo number 2 is maybe in rust, I think so. Yeah, rust only. Yeah, this is another thing. What is the specific fungicide recommended for the management of damping off? Can anyone answer? What is the specific fungicide? Just now we have discussed, right? Metalaxyl. Exactly, metalaxyl. Yeah, metalaxyl. So related to, based on the options, we'll go with generally metalaxyl. Okay, what is the fungi associated with the first physiological work of plant pathology? Can anyone answer? Yeah, Kirti. Botrytis cinerea. Exactly, Botrytis cinerea, that is N20, right? The type of Antheridium. First of all, what is this Antheridium male reproductive organ, right? So the type of Antheridium is produced by Phytophthora. They are asking Phytophthora. Phytophthora? Amphigynous. Very good, amphigenous type of Antheridium. Yeah, exactly. So what is the meaning of Phytophthora? Plant destroyer. Plant destroyer. Who gave this? Yeah, who gave this Phytophthora as a plant destroyer? Yeah. Describe something about Anton de Berry. Point wise, Anton de Berry, lead blight of potato, Phytophthora infestans, 1845, Irish famine, 1.5, around 1 to 1.5 people are killed. Phyto means plant, Thora means destroyer. The life cycle was first, given by Anton de Berry in 1860s. That was identified. In 1876, he named the pathogen as Phytophthora. Phyto means, as I told you, like before naming this Phytophthora infestans, what is the name of the pathogen? Botrytis infestans. Who gave this pathogen as Botrytis infestans? First one is Montag, second one is Berkeley. Who is this Berkeley? Is a British mycology who actually fought for the lead blood. There is a story I will tell you in the beginning classes. That's why I have given the first class is introduction to plant pathology, how this plant pathology started. What you study in your books is different. The story what I am going to tell you is different. Of course, everything is right only, but I will tell you the exact detailed story. From the story, you will understand what exactly plant pathology, how exactly the plant pathology started. Anyway, that is different. Okay, in which of the life cycle vesicle formation takes place?
Pythium.
Exactly, pythium. Yeah. Trade name of metaloxyl.
Redomil.
Okay, what exactly if the metaloxyl plus mancozeb combination is available? What is the trade name? Metaloxyl plus mancozeb.
Redomil gold.
Very good. Redomil gold. Yeah. I will ask you one more question. If someone will answer, I can do special thing for them. The question is, there is one metaloxyl which can be recommended for seed treatment also. What is the trade name of the metaloxyl recommended for seed treatment?
Apran ST.
Very good. Apron.
Apran 35
Yeah.
ST.
Apron 35 SD. That is different. Apron. That is very right. Deepika. Yeah. You message me separately. I will help you something. Okay, anyway, next one. Citrus gummosis is caused by? Yeah, Phytophthora, Citropthora. There are 6 pathogens are there. Phytophthora, Citropthora, Phytophthora gummosis, Phytophthora hibernalis. I will show you. Yeah, take a look here. Here, citrus gummosis is caused by Phytophthora parasitica, Phytophthora palmivora, Phytophthora Citropthora, Phytophthora hibernalis, Phytophthora syringae, Phytophthora cactorum. Actually, in one of the research article, it is mentioned that 12 pathogens are associated with. So, but you remember three very important things: Phytophthora Citropthora, Phytophthora palmivora, Phytophthora parasitica. But the majority of the research articles, it is mentioned as Phytophthora parasitica is the prime cause. But in sometimes we do study this Phytophthora palmivora and Citropthora also. But generally, based on the options, you answer there. Okay, anyway, the term symbiosis was coined by? Okay, okay, lemon shaped sporangia is produced by?
Phytophthora.
Okay, pythium produces what kind of sporangia? I'm exactly globos are. I'm asking sporangia, not a type of antheridium. We must answer though by having by knowing the question. Okay, be cautious when you are answering. Yeah, you guys know very well. I have already circulating and circulated. If anything else, please reach us at the given number. Anyway, I am telling you once again, your subscription is a promise. We will never ever make you feel that you are taking up this decision of subscription. So just imagine if you are paying 2,000, or maybe general I'm telling. For example, we are training you 6 months, 180 days. Every single day you are paying just 10 rupees. So 180 days, 180 into 10, around 1800, 1200 is not a big deal. So it is our promise. We will ensure you that whatever you are paying, every single penny, that is we will revert you back in terms of quality information or worthy information. If you still want to proceed with, you can subscribe, or else if there is anything else also doubts, you can please let me know. If there is any doubts, we are happy to